I love that segue to get started. So Dr. Diego Mastroeni, Arizona State University researcher and professor. So lucky to have met you in this lots of exciting research that you've published and with the team there. So maybe we could start with you telling us a little bit about yourself and specifically what you get excited about in your research and what you're up to these days.
Yeah, certainly. So yeah, an associate professor at the Neurodegenerative Research Center at ASU. This is a newer center. I had the luxury of help building the center about 10 years ago, and so it's blossomed quite a bit over the last decade. And my rationale, my reasoning for ever getting into this field of work, like many, is the unfortunate reality of dementia, which my grandfather suffered from, and he suffered from one of the more devastating ones, which is a front temporal dementia, and it's a more kind of aggressive phenotype clinically, and it was my journey kind of started much younger. I was 16 years old, and my grandfather and I we were really tight. And unfortunately, he was going down the path of being very dangerous to himself. My mother found him driving against the wrong way in traffic on 7th Street as she's heading to work on his bicycle. And in another situation, he actually tried to stab his own son. So it was so aggressive, but he wouldn't leave the house with anybody because he didn't trust anyone. But even during his darkest days, we still had this really tight connection and bond, and he still remembered me. I had the unfortunate task of actually taking my grandfather into the institution. I had to tell him that we were going to do lawn work because he owned a lawn company back in the day, and I had to do that at 16 years old because he wouldn't trust anybody else. But I knew I had to. I had no choice. It was that or something really bad could have happened. So I had to take him in, and it was probably still the most difficult thing I've ever had to do, take somebody in. But you know what's best for them. And that was my initial motivation. And so I wanted to go to medical school, and I wanted to be a neurologist. And I did all my undergraduate work and in preparation to go to medical school, and I wanted to do a rotation in a lab. And that's where I started over at Banner Sun Health Research Institute at the time. And it was an incredible place. And I think we'll talk more about it. But yeah, it was an opportunity for me to learn a little bit more about the neurodegenerative diseases and et cetera. And I got to the point where I was like, I think this is what I want to do. I think that I don't want to treat individuals with medication. I want to be able to identify why this is happening to them, So I found more passion in the research angle than I did, as I was doing clinical rotations with some physicians. I realized that I wasn't nearly as passionate, and I couldn't, that. So that's kind of the rationale of why I did what I did and how I ended up where I am today. And I'm still as passionate today as I was, 20 years ago.
Wow. Well, thank you for sharing that, and to have that perspective clearly infuses your work, and I'm sure helps rubs off on your trainees also and the students that work with you. So I was looking at your bio, and epigenetics seems to be all over that. So maybe you could talk about that and then how that leads to this study that we're going to be talking about today.
Yeah, sure. Yeah, I did my PhD in neuroepigenetics, and basically what that is the ability to, how the environment can regulate our genome. And so I think one of the most interesting thing that people don't really understand is that just because, mom and dad gives you gene X doesn't necessarily mean that, you're destined for disease, let's say. A lot of these genes need to be turned on and off. And that's where this concept of epigenetics comes in, which literally means above the genome and its ability to regulate the genes. So there's a lot of environmental factors that can contribute to, whether some genes are turned on, whether some genes are turned off, et cetera. I can give you a study we published in 2011. We were looking at monozygotic twins that were discordant for Alzheimer's disease. So these are individuals who are genetically identical, and one of the individuals got Alzheimer's disease and the other didn't. So if it was purely in the context of genetics, how would that be possible? Probably should be, but that wasn't the case. And at the time, it took me like almost 10 years to get this paper published because no one even believed me. But now epigenetics is like, through the roof. But
That's hot. Mhm.
that's just pretty much how science works. But what we found out is that both of these individuals were PhDs. They're both chemical engineers. One of them worked in an office job. They lived in the same community, had kids, etc. And the other one worked at a DDT plant. And for those who don't remember what DDT is, but it's a noxious chemical that we use to eradicate mosquitoes and things like that. So it's a pesticide. And so with this exposure, that was the major difference between the two of them, even though they lived in the same communities, etc. And so then we did some genetic testing and looked at DNA methylation levels and a bunch of these genes that regulate, that help regulate neurodegeneration, inflammation, and aggregation of these toxic buildup of proteins, amyloid and tau, the kind of the hallmarks of Alzheimer's disease. These were all upregulated and demethylated, which that would now allow those genes to be turned on, where his identical twin, they were all turned off.
Mm.
And so that's really a focus of ours for a long time is this epigenetics and the way that the environment can influence our genome. And I think that's even timely and really important, considering that now we're looking into all kinds of things in the context of the environment, and one being the microbiota of our gut.
Hmm. Great. So that's such a striking example that you found with these twins who both seems like really lived the twin life. Wow. And I would say, I learned so much just preparing for this conversation. I've been someone that would see these talks on the brain gut axis and sort of just, quickly walk past it. And even though I try to say that I'm open-minded and holistic, I'm very brain centric. And clearly, there's a lot of pushback on this field for how focused they are on amyloid and tau in the brain. And so this was so exciting, and I'm really I can't wait to hear more about so more about like the study and then what you found.
Yeah. So, I think before I tell you about the study, I think that, I think it's important just to set the stage that, people really just don't realize the importance of the microbiome. And the microbiome is just is a whole conglomerate of viruses, bacteria, yeast, fungi. It's just a huge colony. And people don't really realize also that we have more of those in our body than our own cells, right? So, at the end of the day, you start to ask yourself, who's really in control here, right? So if you don't feed your microbiome correctly, what happens? You get sick, you get upset stomachs, or if you're anxious, you get the vagal response or anything like these. These are all factors that are regulated by the microbiome. And even in women, like if we talk about estrogen, in estrogen, we know that women who have dementia are 5-6 times more likely to get dementia. Women are at higher risk.
Mhm.
And the microbiome, the bacteria, are what break down estrogen into its
Mhm.
usable forms. And so we're about to publish a paper here in the next. We just sent it to Alzheimer's disease and dementia. We identified 7 unique bacteria that are identified in women with dementia, that aren't in normal individuals, normal women that do not have disease. And it could be as simple as, giving them, these women, a cocktail of probiotics that would, eradicate either this specific, group of bacteria or whatever. And so that fact, I mean, and also, I mean, and you as a scientist, I mean, and a physician, that that the microbiome is actually responsible for all these all metabolizing all of our neurotransmitters and making our neurotransmitters, serotonin, acetylcholine.
Mhm.
These are all processed in our gut, taken through the vagal nerve and the parasympathetic nervous system, and then then we're utilized in the central nervous system. So it's incredibly important, and I think that I think it's pretty clear now that its involvement in not just neurodegenerative disease, but even in autism. I have an incredible colleague, Rosie at ASU, who's doing fecal transplants in autistic children and is having incredible success. So with that in mind,
Wow.
I started this study in 2020 with Rosie, and we were looking at just the microbiome. And a colleague of mine, Dr. Ben Reedhead, he's a bioinformatician, an incredibly talented bioinformatician. And so we generated all this mass spec data, all this proteomics data on the gut, the transverse colon in Alzheimer's and normal individuals. So we're matching individuals who have disease and individuals who do not have disease. And
Can I just ask a
yeah,
clarifying
shoot,
question
shoot.
real quick?
Yeah, shoot.
So I just want to, so
Yeah,
microbiome covers more than just the gut. It's the whole body, including like the skin and the mucus, everything that, and so I just want
correct.
to make sure that's clear. And because even the, influence of air pollution now with risk of Alzheimer's, that's become established. So that's, coming in through. And so
Exactly.
I just wanted to mention, and then for this study, you focused on the gut microbiome.
Correct. We
Yeah.
focus on the gut microbiome for a lot of reasons. One is that's because it's where it's mostly enriched. And a lot of these processes that affect the nervous system are actually done through the microbiome in the stomach.
Got
Uh,
it.
but yes,
Okay.
you're absolutely right. It's global. I mean, it's everywhere. It's in organs, you name it. It's everywhere, even in just flowing our peripheral system. Um, but yeah, so we started there. We wanted to take this like 10,000-foot view and treat Alzheimer's disease not necessarily as a brain-centric disease, but like a systemic disorder. And we just asked a simple question. So we profiled over 40 different organs and asked like, what is going on with the whole body wide? And the most interesting findings really turned out in the gut. And like I said, a colleague of mine, Dr. Reedhead, he took this data and identified that the human cytomegalovirus, a specific virus, was found in a subset of individuals who have specific genetics. Danny and I were talking about how can I clearly articulate this in a way that it's not like over everyone's head, because it's often over my own head. These bioinformaticians are like magicians. And so I think that the simple way to explain this is that if you have a set of genes that make you at risk for the disease, and you also have this virus an active infection of this virus, your risk of Alzheimer's disease is significantly higher than those that do not. And so what was so exciting about this study was that the possibility of us introducing something as trivial as antiviral therapy. So this is something that's already FDA approved. So if you fall within this subset, which we think is roughly around 35% of Alzheimer's patients, if you fall within this subset, and we're working right now on a biomarker. So what we're trying to do is we're trying to identify a biomarker that we could select these individuals based on, this set of criteria. And that's what we're really pushing on now. We're pushing pretty hard. I mean, we have multiple groups trying to figure out what would be the best biomarker. But if we could identify, we could do something as simple as a cyclovir that people take all the time for cold sores, that would reduce that risk that we're talking about. So I think that's kind of the really exciting part of the study. And I think that, there's a lot of momentum. And so, yeah, I mean, it's everyone's pretty excited about this.
So if I could jump in. So I know that other viruses and pathogens have been linked to Alzheimer's disease. So from what I knew and doing some research looking up before this, so HSV, herpes simplex virus type 1, your study, the HCMV, then also the bacteria or the associated with gingivitis.
Yeah,
So
gingivalis. Yes.
yeah, gingivalis, and then chlamydia pneumoniae has been linked. So those are both bacteria, those last two. And so just for the listener, so if you could just break it down briefly, like, so you had these 100 subject samples from the brain and body donation program, and then you found, if you could go back to that part.
Yeah.
Yeah.
Yeah, so we had a hundred subjects from the brain and body donation program. Again, they were well matched for age, sex, gender, postmortem intervals, all these different things. These covariates that are really important in the analysis.
Mhm.
Um, and so taking the gut samples, right? This is where we first identified this profile.
Mhm.
And then my colleague Ben, he took all this data with a company called Seramune.
Yeah.
We were looking at infections. So we found HCMV in the gut. So then we took their matching samples and we were looking in their blood.
Mhm.
And then once we realized that they have an active infection, we then started thinking about, well, shoot, could it be in the brain as well? And so then
Mhm.
we took these same samples, these 35% of these Alzheimer's disease cases that we found
Uh-huh.
this to be true,
Mhm.
and we identified them. We identified this in these same individuals by histology. We found that the HCMV virus is actually in the brain as well,
And
which
is
is,
it sorry, go ahead.
yeah, no, go ahead. No, shoot.
So thinking about, from what I know, this virus, most people acquire during life and like clear it, so to speak. But then some people it's maybe you'd say latent, and then does it become reactivated at some point? Is that what?
Correct. Yeah. So we don't know that for sure, right? I mean,
Okay.
this
Oh,
is empirical, and I think that some of the next steps in our work will tell us that. But I think that it doesn't really go away.
mm,
I think that they sit and wait. Uh,
yeah.
they're opportunistic, just like
Mhm.
a lot of bugs are. And so they're just waiting until the immune system, and this is where that IgG4 stuff comes in. I just didn't want to, talk too much about the immunology because immunology is really complex, but IgG4 is one of the factors within this subset of individuals. And what that does is it actually dampens the immune system. And by dampening the immune system by these individuals, that's one of the variables in these individuals. So you have genetics, which is that CD83 specific subtype. You have this IgG4 profile, which suppresses the immune system, and you
Mhm.
have active infection of HCMV. So this three-prong hit, and so that's kind of where we're at. But so we identified it in the brain. And then we asked the question, well, how is it getting from the gut to the brain? And that's how we went through the vagal nerve, which is a direct route from the gut into the central nervous system. So that's kind of the pathway that we took.
Got it. So from my reading, so there's tell me if this is how you understand this. So, and these are some things I didn't realize. So amyloid beta is plays a protective role at first as an antimicrobial peptide. And then it seems like from chronic infection and then maybe the gut imbalance with the microbiome there, then you have this immune activation. And then with AD patients, there's like increasing data that the gut microbiome is less diverse than in people who don't have Alzheimer's disease. And then also just there's more inflammatory factors in the gut. And then so this leads to this, I guess, transport. Then it goes to the brain via the vagal nerve, and maybe the brain is also there's this immune activation and pro-inflammatory. Maybe the blood-brain barrier is more leaky for all that. And then there's more amyloid beta. So it's like this vicious cycle. Is that
I
kind of
think, yeah, I think you're right on point. Yeah, I mean, look, this is hypothesis driven, right? I mean, we don't
yeah,
really know
sure.
exactly how this is happening, but I think that's a solid argument, right? I think it's just a significantly pro-inflammatory environment.
Yeah.
And this stuff isn't really new either. I mean, you know, uh, somewhere like 1909, Alias Alzheimer's first described um, that he thought that this was due to a viral infection. This is a
Mhm.
century ago. Uh,
Yeah.
I mean, this isn't really new. It's just, I think we just now have the technology to really start asking those kinds of questions that just wasn't available even 5, 10 years ago. Uh,
Mhm.
so yeah, but I think that your stepwise fashion, it makes sense, right? I mean, we know that amyloid is antimicrobial. I think it's just so many things. Like no one really knows what it does. Like they know it's associated with a ton of different things.
Yeah.
When I look in the brains of Alzheimer's patients, most cases, it's always associated with a blood vessel. So it,
Mm,
it makes you think, is this really something that's either leaking out? Is it stopping things from leaking? It's stopping things from coming in. It's mopping things up, and then you just get so much of it.
mm,
It's very difficult. But in our study, what I think is worth pointing out is that we did brain organoids, and this is a way to try to recapitulate the human brain by growing these three-dimensional brains in
mm,
vitro, and we treated well these brains have they overexpress genes like APP and tau, and
mm,
so they're making a lot of amyloid and tau, the two hallmarks of Alzheimer's disease, right? And then
mm,
we infected the organoids with the HCMV virus. And what seems to happen is that you get an increase in production of this both increase tau phosphorylation and amyloid aggregation. So it
mm.
has a direct effect, and I think that was something that was really exciting about our work because a lot of the stuff that's out there so far doesn't they haven't really been able to show any effect, let's say a causative effect. But
Mm.
like so I think that's a really exciting part of our research that it does actually increase in fold the aggregation and the phosphorylation of tau, which is like I said, the hallmarks of the disease.
And I guess what that made me think about was, there's like it's like this slow moving train that suddenly like speeds up. You can kind of see like as those things are being produced like all together, like we know that once like the tau hits, then the symptoms start. So clearly there's so much else going on that we haven't even measured yet or can't yet with our technology. So um I was curious about the clinical symptoms of the people, the subjects with Alzheimer's. Did that, did you see anything there that was like I guess it's difficult because it was a small sample and like, did more people have neuropsychiatric symptoms, for example, or you can't tell with this small of a sample?
Yeah, I think that's a great question because, Ben, he's actually doing all that workup right now. These people have a lot of data on them, and I think that's one of the most impressive things about our brain bank, at Banner Sun Health Research Institute is they're followed longitudinally over a long period of time. That's a powerful job for a biostatistician, definitely
Yeah.
not for me. But yeah, I think that's a really good question. And they're looking at all kinds of stuff besides that, like hematocrit, looking at there's too many variables. But yeah, they're doing obviously the mini mental state examinations, the MOCA. They're doing all your standard stuff. But like, I mean, I think maybe you would be an important factor maybe in looking through these data sets in the psychiatric end. Um, I think like I was saying about the brain bank, and I think what's so important about it is that it's such a sacrifice for these families to donate their brains and body to research. Um, and I don't think anywhere in our career, in my career, in my colleagues' career, at least in the state of Arizona, and globally for that matter, would any of this be possible without having these incredible subjects and their families that are willing to donate to the brain bank, which I think is probably what sets us apart from anywhere else globally.
Mm.
And so that is something that I'm really thankful about, and I really want to focus on, and also I think another interesting part of the study is that most everyone in this study is all Caucasian, and
Mm.
and usually from the Midwest. Um, it's because a lot of the people from the Midwest, they migrate over here, and I think that inclusivity is really important, especially in the context of neurodegenerative diseases, where we even see higher risks of degenerative diseases in Latinos, for example, and even in African Americans. But the issue is that they don't really have a long-standing good relationship with research. So it's very difficult, and I think that's something that needs to happen in order to get a more global perspective of these neurodegenerative diseases. Because as of right now, we're really focused on a subset of individuals. So I think that's important to point out.
Oh, absolutely. And just to step back and just give a basic overview of what these brain and body donation program, this one at Banner in Sun City, and these are about throughout the world, is that people, so for the local one, 18 and older can sign up, right? And so they get these yearly, a whole bunch of cognitive, functional, mood tests, questionnaires, and then neurologic exam, movement exam, right? And then there's different kinds of maybe brain scans, lumbar puncture, CSF testing, cerebrospinal fluid, if they're open to that, blood probably now. And then when they die, they agree to donate their brain and or also their body. That's an option too. And then so many discoveries have come from that, and it's been going on for like 30 years or something. Is that right?
Right.
But yeah, they're making some progress generally with non-Caucasian populations. But we know, and I was going to ask this about your study, but we know that Alzheimer's risk gene ApoE4 seems to play less of a role in the dementia associated with, different non-Caucasian populations. And so, but we don't know enough yet because we need more people to get involved. And there's, of course, many reasons, like you said, that people don't yet. But in terms of, I was curious if you found ApoE4, it sounds like, ApoE didn't
It wasn't a
show any
strong
difference.
association, no.
Yeah.
Not in
Okay.
these individuals, but again, we're talking about 100 people. So I'm in the process of doing 200
Yeah.
more people right now. So
Great.
so we already have the serum immune data out, and I've already we're doing histology on 200 more brains,
Mhm.
and see what this data pulls out. I mean, it's one thing when it's 100 individuals, but when it's 300, you start to like, okay, well, look, we got a pattern here. I'm sure it's going to sway. It'll there's always going to be ebbs and flows, and just by population genetics. I mean, if you're going to have the correct number of individuals within that cohort, you just don't know. I mean, it's arbitrary. Like I don't know how many CD83 individuals are going to be in this cohort. We have to test it genetically. And so if you have a really small portion of that, obviously that's going to be less, but what other, I mean, we have to test everybody. So the idea is to test all 2,000 people in the brain bank. But we're starting a little bit at a time, as funding trickles in, we'll continue this, we'll continue as fast as we can go. But yeah.
And you validated this in another cohort, right?
We did. We validated this in another cohort at Rush Medical Center, and so we used the RoseMap data set. I don't know if you're familiar with that. It's these are nuns.
Oh, okay. Yep.
Yeah, it's a religious order nun group. Yeah,
Yeah.
yeah, and they're followed over very similar to our studies longitudinally over 100. They're like what, I don't know, 40 years old to 100 years old or something like that, or even older. And we looked at an additional, oh shoot, I can't remember, was it 400 samples or 200 samples? And we found very strong an association, correlation between the two groups. So that was also additional validation, something that we were really excited about because, brain banks do things differently. And just because you find it in one brain bank doesn't necessarily mean you're going to find it in another one because they preserve things differently. And but yeah, it was pretty exciting to validate that in a larger cohort.
Cool. That's great. So thinking about treatments, potentially, of course we all want, like you had mentioned, like could antivirals, for example, and there's probably people already going on acyclovir, like after they saw your study,
Well,
but more and more I'm thinking about, well, I have these patients who are like the picture of health, and yeah, they're older, they're like in their 70s, late, even early 70s, say, but they're getting Alzheimer's disease and they're not ApoE44. And so it's like they did everything right. So it kind of makes sense that there's something like this that like they had no, like they couldn't do anything about that if they had this like genetic makeup and they had this active, infection. I just, I was thinking about some other things and I know we're all speculating right now, but in terms of like, so because there's multiple viruses, bacteria, things that have been linked, it's hard to know like, you know, what exactly until we can measure. Like not everyone's going to have like maybe a virus that responds to acyclovir. Maybe someone has like the gingivalis, you know, for example. So this is tough at this point, but things like probiotics. And then I saw, you know, some fecal material transplantation talk, and I know that's more talked about farther along in the Parkinson's space. And so I'm just curious about your thoughts on those things.
Yeah, you know, I mean, these are just thoughts, okay? So, yeah, so, yeah, like probiotics, for example. I mean, I think they're okay. I think it has to be more personalized, though, right? And so, if you don't know yourself, your own microbiome, for example, I think that would be a really smart thing to do first, is really maybe profile your microbiome. And I know there's places out there that do that. I think that would be more advantageous.
Mm.
Um, I think that, you know, taking a cyclovir on a whim is a bad idea. I think, you know, I think if you have an active infection of a cyclovir, which is something you could really easily do, right? And that's kind of like where I think that the inflammatory system is really responsible. And I kind of think of it as more like an accelerant, right? So like pouring gas on a fire or something like that, right? And it really drives, you know, your maybe underlying just sleepy Alzheimer's to a very aggressive up front dementia. We don't know that though. I mean, but I mean, it's something to consider. I mean, is there, you know, I mean, should we be looking at other things like that? P. gingivalis is another one. We just put in a grant with colleagues at Stanford on P. gingivalis. It might be what we were considering multiple insults. You know, the
Mhm.
HCMV
Yes.
is just the one that we've really found, but like we're doing other studies in rubella,
Mhm.
um, and we're finding a ton of different viruses that are going kind of crazy. And it might just be the inability to maintain self, you know, your inability to recognize viruses as we're aging. And it just may be just a whole host of these bugs, you know. Um, and it doesn't matter which bug it is, as long as we can identify a subgroup of individuals that we can help treat. You know, I mean, it might be P. gingivalis for this subgroup. It might be HCMV or HVV6 for another group. You know, it's, you know, we'll have to wait and see. But yeah, I think it's good we're having the discussion. And I think that moving away from brain-centric Alzheimer's disease, I think is really good for our field. I've been brain-centric for 15 years, and the last five have been, you know, I've been open to the possibility. You know, I'm new to the cause as well.
And I would, yeah, thank you. And I'm thinking about the timing of treatment and, you know, when I was at Banner, we did trials on the people who had the gingivalis, you know, whatever antibodies are, and we had an HSV1 study
Mhm.
for treatment for people. So, which I think was with acyclovir, and both of those I believe were negative phase 2, but clearly it's too late at that point. I would say it seems like, if you know, do you agree with that? Like it probably, I would think, you know, midlife or maybe even younger. Or can you speculate on that, or is that?
Yeah, no, I mean, I can speculate on everything. It's my job. But yeah, no. So I think if you started something like this in your 30s, I mean, even one round a year would reduce
Mhm.
significant risk and really dampen the immune system to not be so overactive. Um, so I think it really is going to be a really special subset of individuals, and I think that's kind of what we're excited about in this project, is that we're really focusing on a group of dementia patients, not the whole, you know, 100% of individuals. And I think that's just really the way it's going to play out. I think there's going to be like maybe 20 or 30 different little subcategories of Alzheimer's disease or dementia, you know.
Oh yeah,
I just don't think
at
that
least.
one shoe fits all here. And so if we can help even 1% of the Alzheimer's population, I've fulfilled my duty as a scientist, and I can sleep at night forever.
Yeah, wow. Yeah, that's a lot of people. So the subset, is that the CD83
Correct.
positive people, or what
Correct.
do you, okay,
Yeah, so those are CD83
got
positive
it.
and IgG4 positive
Okay.
and positive for the immune, their immune system. Yeah, I mean, that's what I'm saying is that like that's why we're looking for a biomarker because we're not going to do all that. It's impossible. That'd be like tons of money and no one,
I see.
yeah.
Yeah, got it.
What's good for the heart
Yeah.
is good for the brain, so that's always easy. But yeah, I think that there's a lot of anti-inflammatory foods that you can look at, that I think that are really rich. I think living on
Mhm.
some of these Mediterranean diets is not a terrible idea at all. I think, it's very reasonable.
Yeah, and then it's challenging to talk about doing any of these things in younger people because there's still such a stigma about Alzheimer's disease that a lot of people maybe wouldn't want to step forward, to delve into that if it's not clear that whatever they're getting is going to be definitely helping. So,
Yeah,
um, it's a challenging area.
yeah, it's tough.
Anything else before I was gonna take some of these
Oh,
questions?
no, I think that's that's the story. I'm sticking to it.
Yeah, great. And then Grace asked, I'm curious whether correlations while the individuals were living have been made with MRI studies, and were there protein markers for 20 years prior to their pass, or like building up for 20 years prior to their passing?
No, there definitely isn't. The proteomics in terms of like the new tau stuff that we're doing now for the biomarker stuff has only been around for the last 3 years ish. So we don't have a ton of data of that as far as like longitudinally. The imaging stuff, some of these individuals have MRIs, but honestly, at the end of the day, it doesn't really matter if they have MRIs because we're looking at pathological data. So
Mm,
that means the neuropathologist looked inside of the brain and confirmed that they have Alzheimer's disease. So MRIs are they're good, but neuropathology is the gold standard. And that's kind of one of the things that we know about, even clinically defining Alzheimer's disease is, one of the things that's really tough is to fit you into a specific category. And then it's not until, you have a pathological diagnosis that you can be 100% certain that your clinical assessment is actually, reflective of the pathological assessment.
got it. Um, if anyone's interested in learning about the brain and body donation program here or wherever you're at, reach out to me. I'm happy to give you some information. Okay. So thank you so much for your time and insights and for what you're doing. And I'm so excited to learn about I mean, I'm sure you said it's gonna take a long time, but when do you expect like the next phase of findings to exist?
Yeah, I hope it's actually within the next year.
Oh, wow.
Yeah, I
Awesome.
think we're pushing pretty hard. We're even starting to trying to push a phase 1 clinical trial with cyclovir. We'll see how it turns out, but
Yeah.
I think it's worth a push if you have the virus active. Um,
And what population for a phase 1 trial?
yeah, it's a good question. I don't know. I mean, your guess is as good as mine. I mean,
Like,
I think you have to be like mild cognitively impaired. I think that I think that and even aged without dementia or out cognitive impairment. I mean, I think you might have to span the gamut. I don't know. I'm actually apprehensive, but these guys are gung ho about clinical trials.
so, there's more and more talk about developing risk profiles,
Yeah.
especially for Alzheimer's risk. So that's people who don't yet have symptoms. And so in thinking about who's going to benefit from a treatment, I, I would actually say that they'd not yet have symptoms, but there's enough biologic and risk data to support that they're likely to develop symptoms of Alzheimer's disease like in the next
Yeah,
10 years, which we don't have that all delineated, but
you get an idea.
like yeah, but like say they have elevated amyloid and elevated tau, but
Right.
and maybe they're ApoE4/4,
Oh
but
yeah.
they're asymptomatic and they're CD83. Like, yeah, well, I'm excited for that. I look forward to hearing about that. Awesome. Well, take care. Keep us posted.
All right, Danny, it's great to talk to you guys. Thank you guys very much.
Thank you. Thanks, everybody. Have a good day.